All right, today is September 11 in 2025, and my guest is Andrew Hessel. Andrew is a pioneer in synthetic biology, futurist, microbiologist, and geneticist, inventor, and serial entrepreneur. Andrew, welcome to the show.
Thanks for having me, Nick. Good to be here.
Yeah, so let's jump right into it. Andrew, what is life?
What is life? That's been a question that a lot of people have uh, tried to figure out. I look at life personally as near magical chemistry. Biochemistry is near magical chemistry, uh, and it's more than just near magical chemistry. It's near magical physics. Biophysics is completely different than the physics of the everyday world. Today, we can map the position of every asteroid, planet, satellite with perfect precision because the rules of the physical universe are really clear. The formulas are clear. The math works. Biophysics is near magical, and we're still trying to figure out a lot of those rules. We're still trying to build the environments, computational environments that can simulate it. And so combined, biochemistry and biophysics is really it creates the architecture for an incredible machine of the living cell. And of course, living cells comprise all living things.
Yeah, and why is that? Is biophysics just more complex than the physics of gravity and things like
Well,
or
it works at a scale, this nanoscale that is unfamiliar, and where even even the motion of atoms and molecules at that scale are dynamic. They're constantly in motion, and so we don't have the same level of resolution and certainty understanding molecules at that scale of matter. And of course, everything is in fluid. It's constantly dynamic. It's floppy. And so it's just a really complex problem. I am hugely optimistic that we will one day be able to pull up a virtual window and just throw in molecules that we've deciphered, and that in this simulator window they will do the same things in silico that they do in the natural world. But we're not there yet. Drew Hall, the molecular animator that has produced some beautiful movies of kind of the inner working of cells, literally has to hand animate these molecules, to show their activities. I'd love for all this just to be, just another simulation that we run.
yeah. So let's have that conversation, right? Where we understand how we got to where we're now and what we know and what we could potentially, or what do we need to know to do different things in the future, more things, better things with synthetic biology. Maybe you can walk us through a bit what we know now, arriving at kind of the puzzles we're facing through the lens of your own career. How did your thinking evolve, and how did you learn about synthetic biology and life and biochemistry and biophysics?
No, it's great. So I describe myself as being a geek. I loved when I was a kid. I wanted to know how everything worked, and my parents would buy me these books, how things work, elevators and machines and pulleys, et cetera, et cetera. So I want to learn how everything worked, and I took everything apart that I was ever given, every toy, every radio, et cetera, et cetera. So I love that, and so if it was computational, mechanical, electrical, yeah, I had to see the guts of it. And I was actually, because all that comes together in automobiles. I loved working on cars, and I actually figured for my career I'd end up doing something in automotives. But one day I woke up just before I had to kind of lock in my programs, and I basically had I don't know, a voice in my head, and it basically told me to go and study biology. And I didn't. I've always trusted those types of commandments from my subconscious, so I did. I switched my whole program to science, and as soon as I got into microbiology, it clicked. My very first classes in university, because in microbiology 101, in the very first lab, they gave me a whole bunch of petri dishes, and swabs, and they basically said, go exploring for microbes, and you swab your mouth, you swab your skin, swab the top of the fridge, whatever. And the next day you see all of these microorganisms growing on the petri dish, and I was hooked. And I realized, okay, I want to understand how these invisible things work, how single cell creatures work. I immediately saw that they were molecular processors, so very similar to the computer processors that I was working with, and that they had an operating system written in DNA, very similar to the operating systems of my computers. And that was it. After that, I wanted to understand the mechanics of the cellular systems. I wanted to understand the code, and ultimately I wanted to be able to read and write that code and be able to program them. And that's been the low level, guiding star through my entire career.
Mm-hmm. Great. And how did at what pace did our knowledge, especially of the human genome and genomics, evolve, right? So it seems that there was a massive acceleration the past couple of decades, kind of starting with the Human Genome Project. And kind of now we're getting into territory where we, develop therapies and vaccines that use gene editing, that use mRNA technology or different delivery mechanisms. So at what point are we now? What are the things we can do or can master?
Well, it's been a long process. Like I started learning cellular and molecular biology in the 1980s. There were no databases of code at the time. My first copy of GenBank, which is the public repository of genetic code, came on floppy disk, if you can imagine. And the next year came on CD. And after that, it was completely online. It was just too much information. Today, GenBank has about 44 trillion base pairs of code on the last release, and really summarizing thousands, tens of thousands, hundreds of thousands of different genomes, microbial, plant, and animal. So, and of course, much of the growth of these genetic databases came from, as you said, the Human Genome Project, which took years, like 6 years to kind of launched in 1990, and was scheduled to take 15 years to complete at a cost of about $1 a base pair. It was budgeted for about $3 billion, which is like 7 billion in today's money. Over 60,000 Bitcoin if you're into crypto. so,
As many
so
of
it
us
was,
are.
yeah. So like it was a huge chunk of money. And it was made this big life science project, but, that was the catalyst for getting to this. And the important thing about reading genomes is it's kind of your digital microscope into an organism, and it gives you the operating system. So if you can read it, it is non-ambiguous, it is amenable to computational analysis, and it gives you, and if you're comparing, it allows comparison digitally as well. So now you can look at an organism in health and an organism in a state of disease. So it is a huge resource for understanding an organism, but also for doing diagnostics. Is something present? You can find it with the code. Is it broken? You can find it in the code. So that's had a massive return on investment since the Genome Project was complete, and it's powered most of human diagnostics, of course. And then, following in the wake of that work, should have been synthetic biology, our ability to write DNA, and it has. It's just moved a little slower because after the genome was complete, the human genome was complete, the all this scientific effort that had been unified on this one project fractured into 1,000 omics, proteomics, metabolomics, etc. And it's the synthetic biology, which is our ability to code DNA and thus biology, has moved relatively slowly compared to all the effort in sequencing, but now it is accelerating. So if reading DNA code can help in diagnostics, writing DNA code opens the potential to completely program, reprogram, repair, and essentially cure every genetic disease.
So I want to talk more about both reading and writing DNA. And let's start with reading. How does it actually work? I think I really don't know. I never asked that question. How does genome sequencing actually work? Wow, what got us to the point that we can now do that for extremely low cost compared to like millions that it costs to sequence a genome?
Yeah, well, the technology has been evolving since it was first invented. In the early 1980s, it was a very slow process that required some pretty elegant bench work in terms of the reaction. To detect what was happening, we used radioactive radioisotopes, and you'd have to run the sequencing gels on these huge polyacrylamide slabs. It was not a fun job, and it took a lot of work to sequence just a few hundred base pairs of DNA. Ultimately, the reactions got simplified. Instead of radioactive isotopes, we started to use fluorescent dye markers, and the entire process became automated and multi-channel in starting towards the late 90s. It was really accelerating, and it got to the point where you could just load plates that had run the sequencing reactions onto robots, and just they would run, and all this data would stream into the computers. Today, some of the most elegant sequencers are nanopore sequencers produced by companies like Oxford Nanopore. And what they essentially are is a little protein donut. And as the DNA molecule goes through that donut, it blocks a bit of current. And we're talking extremely small currents and amounts of electricity. But the electronics are sensitive enough that as that string, that chain of DNA goes through that donut, you can directly read the bases of DNA by the amount of current they block. And all that data streams in. So now, instead of having you can have thousands, even tens of thousands of these little protein donuts on a membrane, and get streams of many DNA molecules, and all of the analysis is moved to the cloud.
Yeah.
So and today I'm seeing next generation systems that are even more sophisticated, with some of the enzymes that are used for what they call sequencing by synthesis, directly attached to electronic chips. And instead of using kind of these protein donuts, now we're getting some solid state donuts that can be made with, again, many of the precision nanofabrication tools that we might use in chip making. So I expect that DNA sequencing, which has already dropped like 6 orders of magnitude in price, will continue to drop and become more accessible.
Yeah. And at an even more basic level, what is kind of actually the language that the code is written in? And how does the code kind of function, right? So we know that it's not simple if-then statements, right? So where you can read like from one base pair to then this will happen, right? There are like some simple like monogenic traits or diseases, sure, where you can like very precisely isolate where some instructions in your body are coming from. For most things, that isn't the case, though. There's things like polygenic traits, and it's like a complex set of algorithms between different set of instructions, right?
So this is I have a lot of conversations about this, because I work with single-celled organisms most of the time to be able to do precision work, because single-celled organisms don't have well, let's just say they're billions of years less complex than we are. But the code is digital. This is something that most people have trouble grokking. Like, we're used to digital information in our computers, and we know that all the software and all the tools we use are ultimately manipulating zeros and ones in the form of electrons. That's electronic computing. With DNA code, it's also digital, but it's a molecular code. It's like molecular links in a chain. But there's only 4 types of links, which we represent by A, T, G, and C. And so it's actually a more compact and concise digital language than zeros and ones. So you can encode a lot of information when you have base 4. But that digital code, it's the executable. Like when we write computer code, we write in a human-friendly language today, and a piece of software called a compiler turns it into code that the processor can run. With DNA, we're looking at the executable code that the cell can run, and we have to reverse compile it into a form that people can understand. And in fact, today, the best understanding is actually the AI tools, the AI scientists that we've been training on this language set. But we can identify on and off switches. We can identify what codes for a protein or a metabolic pathway. We've done the reverse engineering on that executable code so that we have a pretty good understanding of what most of the code is doing. But it's vast. And as again, if there's a single base pair error in the code, it's a little glitch. But that little glitch might be in just one protein, but a protein that's so important that ultimately it causes a really serious disease like cystic fibrosis. Or we can have, but it's also, to remember, it's spaghetti code. It's not like any person logically organized it. It's what's evolved over billions of years, and it works, but again, it's not always logical or rational. And there's a lot of interaction with code across our genome, which is why we get these so-called polygenic states, traits, whether it's our features, whether it's how we age. What's important, though, is that as we start, to be able to program DNA with precision, we don't start at the top with humans. We start with simpler systems. We start with proteins and components. We move up to viruses. We've now moved into single-celled organisms like bacteria and yeast, although that's still out of the price performance reach of many academics today. It's not widespread. And eventually, as DNA synthesis and assembly technologies get better, and the software tool for designing DNA code will keep marching up the evolutionary tree to more and more complex organisms. So we are in this incredible transition now of going from understanding the code, reading it, and comprehending it, to now being able to write it with precision. And that opens up a huge application space.
Yeah. So we're going to talk much more about the write part, but still about the read part, right? So one thing I've always been thinking about biochemistry is kind of a system, as you said, a digital system, right? So we have kind of the code for it. But it's much harder to reverse engineer because we haven't created it from scratch, right? Sort of digital language we have created, and we can reverse engineer because we can trace it back to, every human that contributed to the code base, right? And it's executed kind of deterministically through chips and, compilers and code that a human at some point has written or touched or made. Versus with biology, that's not the case, right? So we weren't the engineers of biology. We kind of have to reverse engineer a dynamic, complex, evolved system that, that has built up over millions and billions of years, right?
You've nailed it on the head. Like, biology is the only technology that we didn't create. It created us. And as you pointed out, we have all the stories, all the patents, all the information on every other technology, including everything digital. And it's accessible. With biology, it is a process of reverse engineering, and we've been doing it for centuries, like if not millennia. We started breeding programs millennia ago, and just figuring out how we can manipulate it. But on the actual reverse engineering, we've been cataloging species across the planet, and now as we've gotten down into the molecular, it's just a higher resolution form of dissection. And of course, once we get to the actual genetic instructions, now it becomes easier because those genetic instructions are conserved across every living thing. There's, as I started out talking about biochemistry, the biochemistry of cells, any cell, from a bacterium, plants, animals, all the way to us, is highly conserved. So it's basically the same core processes in all living things. And they use the same conserved language, genetic language. It's not like every organism has its own genetic language. We all use a standard code, which is pretty remarkable. And it allows us to do things like take code from a human and put it into a bacteria and have it express a human protein, and vice versa, take code from a bacterium and move it into a higher organism or into a different organism. It's
Mm.
pretty wonderful how much we can mix and match.
Yeah. How much do you feel you, as someone who's been in this for decades, and by extension, humans know about biology, kind of compared to other systems, right? So if I look at like computer engineers, there's lots of things you don't know, but other engineers know, and it is in books simply because it was created by us, right? So kind of the limits of your knowledge are the kind of things that other humans know, versus when you kind of look into the stars and we're realizing we're one small fraction of like a bazillion of different stars and universes. We're kind of realizing, okay, that's unknowable to us. No single human doesn't even have the fraction of a knowledge of like what the set of universes or the universes consists of. And that spectrum, how does it feel like in biology? Like, does it feel like we're getting to the frontiers of what's knowable in it? Or does it feel like a vast universe?
It feels like a vast universe that we've barely scratched the surface of. But because, you can think of each organism as its own universe, we are getting, there are some organisms, many of them, what we call model organisms, that we are just putting a huge amount of attention into to understand their function. I like working from the single cell up. And today we've like the E. coli bacterium is one of these model organisms that has helped us unlock the molecular processes of living cells. Another one is yeast, another single cell organism, but about 1 billion years more advanced than a bacterium, because it's called a eukaryotic organism. It means it has a true nucleus, which is kind of like the hard drive where the genetic material is stored. And that was an evolutionary improvement that took 1 billion years past the bacterium. So yeast is another really powerful model organism. And we've with both E. coli and yeast, we've been reverse engineering, learning all the code and the functions of that code for many, many decades now. All of this going into databases. And we've also synthesized both genomes of those organisms from scratch. So we've demonstrated the capability to build them just using code. So that's huge. And it's really the starting point for so much more engineering as we go forward. But even just taking a look at the protein universe, the components in those cells, we can now essentially engineer any protein, which is great. The tools have got us to that point, which is huge opportunities. But even if we could, even if we engineered every single protein that nature has made on this planet, in every plant, animal, or microbe, that would just be the tiniest drop in an ocean of protein possibilities. So we're talking potentials here that are more than the number of stars in the known universe. So we really have to open our minds to the potential of what's possible as we start to engineer these systems, because we're really at day one here.
Yeah, that's what I thought it would be. So let's go now to the right part, right? You said, and that synthetic biology is basically about how we write the code of life, right? So what are the different ways in which we can write the code, and what does it manifest as? What are the things we can manipulate and create in the physical worlds with the knowledge that we have?
The beautiful thing about DNA code, and again, I mentioned that all cells plant, animal, or microbe have basically the same molecular operating system, which is good, the same core processes. What it means is that being able to program any of these organisms ultimately reduces just to the ability to write DNA code. If you write a little bit of code, just a few thousand bits, base pairs of genetic code, you can make a protein. You write a longer stretch of DNA code, and literally physical size of, as you're putting together like links in a chain. Now you can encode a metabolic pathway, perhaps encoding 5 or 10 or 15 enzymes that can take elemental materials and turn them into a complex molecule. You write a little bit more code and you might make a virus particle, viruses to me are not alive. They're like like USB sticks to plug into cells to load a program. They're kind of like delivery vehicles. And if you write a little more code than that, kind of the minimum to make a living cell is about 500,000 base pairs of code. You can start to make a free living cell. And everything after that is just adding more and more complexity. But so our ability to write DNA code today, well, the ability to write it started with the chemical synthesis of DNA bases back in the 1960s. In the 1970s and 1980s, you could only string together very short strings of DNA, 10, 15, 20 base pairs. The most I ever saw was a couple of hundred. And then, but those technologies have continued to advance. And today the companies that do this work as a service, there's still no desktop, really powerful desktop DNA synthesizer, like a molecular 3D printer. There are some, they're very expensive, and they don't make very long DNA. But most of the companies that have built bespoke automation to go and assemble DNA are improving exponentially. But we're still, I think what's offered commercially today is typically under 100,000 base pairs of code, which is still, a lot less than what you need to just print out the genome of even the simplest bacterium. So that being said, the technology is always evolving. I wrote a patent with some professors, 6 years ago that was awarded for a new type of synthesizer. I don't know if it'll ever be built. It'll cost a lot to build it. I'm seeing new technologies constantly appearing that are looking at how can we assemble DNA faster, better, cheaper. And I am hugely optimistic that we can get the cost of writing a bacterial genome down to about the same cost as reading it. Because it's when you think about how much does it cost for one E. coli to copy its genome and become two? Like the processes are essentially free. We just have to learn to tap into those processes and direct those processes using our other technologies. But these will all be chip-based technologies and nanotechnologies that should, at scale and perfected, be very inexpensive. So I believe that we will soon, I'm talking on the order of 10, 20, 30, 40 years, we will have the technology to write almost any genome on the planet, because it will grow exponentially. And that those exponential growth curves are unfamiliar to most people. But when you can go from, they just go like a hockey stick. And most of the genomes on this planet of the most complex animals are still relatively small in terms of gigabases. Humans, mice, dogs, camels, and most of the agriculture, we're under, we're all in the 3 gigabase range. Plants can have very large genomes. Those will take longer. But once you kind of go away from the megabase to the gigabase, the opportunities for programming just opens up remarkably.
And let's help me understand a bit better the link between write and edit, right? So I'm assuming I'm not a scientist. Many listeners may not be, right? So write in its most basic form, given that DNA is basically digital information consisting of 4 letters,
Yep.
can be represented in the most easiest form simply as like text, right? But that's kind of not what we mean when we mean write, right? So we're using the instructions to create things like proteins, potentially viruses, um, maybe molecules potentially, right? So what are kind of the actual things or the physical manifestations of the genetic code or the genetic material that we are able to write and what we use it
Mhm.
for, right? So, which is basically the added part, like we're doing something with it. Like we're injecting it to people, we're giving it to some people as injections or as pills, or there's completely different pathways. The pathways of like what you mentioned, like the 3D bioprinting potentially. So can you help us understand a bit that link?
Sure. Let me just touch on the synthetic part here. Again, I just told you we have the ability now. Let's just call it 100,000 base pairs, where we can literally type on a computer or have a computer design that code as A's, T's, G's, and C's on a computer screen, and then we can hit print and build that molecule. So any editing that we're doing on that code, we can do on using software, and just hit print. So that makes the editing of these smaller constructs for
I
a
guess I
protein or
misspoke a
even a
bit.
virus easy.
I
Yeah.
didn't mean edit. I meant more deploy, right? So
Well,
yeah,
yeah. So
deploy.
yeah,
Mm-mm.
yeah. But as soon as you print, now you've created those instructions. So let's just stick with DNA for a second. Say one of the first things I wrote over a decade ago with my team was the entire genome of a virus that infects a E. coli cell. So we designed it on using software. We sent it to a company to build the DNA molecule, and we got it back in a little microtube. So then we took that DNA molecule and we put it into an E. coli cell. And thankfully, that's a really easy procedure. It's easier than loading software on a computer. You just heat up the cell, it picks up DNA from the solution it's swimming in. So we loaded that genome into the E. coli cell, that new DNA, and it started making virus particles. So because it reads those instructions, those genetic instructions easily. Now, if we were to, now jump into humans, we don't have the ability to completely write a large program to go into a human cell. We can only make small tweaks to that existing code, because that's a 3 billion base pair genome, well outside of our ability to synthesize today. But say we just want to make a temporary change to that cell which is running the program. There we would use a molecule called RNA. It's a chemical cousin to DNA. It doesn't last a very long time. It's kind of like a photocopy of the reference material. So we can synthesize a short string of RNA and either wrap it in a little bit of lipid, a little bit of fat, so it can merge with a cell and deliver that RNA into the cell, or we can put it into a virus particle, which directs it a little more carefully into the cell. And if we do that, now we can load that RNA in, and that RNA is what's used by the cellular machinery to make a protein. So now that cell is going to start manufacturing the protein that we've just told it to make. That's how we made the COVID vaccines. It's how we can do a lot of short-term engineering, transient engineering. It's not going to last a long time on a cell. But if we decide that we want to make a permanent change to the cell, now we can actually load in DNA, and we can use another system, these CRISPR gene editing systems, which allow us to make a little cut and a search and replace essentially into a larger genome. And now we can affect a permanent change into that cell's genetic code. So that would be a gene therapy. And that would be a permanent change to that cellular's program. So we have a lot of flexibility now. We can program small things with digital precision. We can make transient reprogramming of cells that, to make a new protein for a short term, or we can make a permanent change to those cells to affect a repair, for example, or make an enhancement. And these tools, although still relatively early in their development cycle, will only get better. And to the point where, we will be able to change parts of our body cells, because we have 40 to 50 trillion of those cells in each of us, or do it to the embryo, to us, the single cells that we all start off of, to make a permanent, to do essentially genetic surgery on embryos, which would affect every body cell afterwards.
Yeah, and in my role, exposed to much of at least companies that are kind of spinning out some of that research and then using it, for example, to enhance follistatin production, or there's a company here that is doing VEGF gene therapy, or Clotho is something I'm increasingly hearing about. There's companies who are doing genetic selection, which is, I guess, not the editing part, more like the sequencing part, instead of getting a lot of knowledge, especially like on polygenic traits and things like that. There's always a bit of chatter with a bit of, okay, let's not go too far there when it comes to like things like cloning or actually editing embryos, right? So from what I've been hearing, and maybe you can tell us more, there's a lot that's possible there, but we're not doing it because for ethical reasons.
Yeah, so in general, only working with humans kind of pushes the barriers with ethics. We have a lot of flexibility when we're dealing with model organisms, even primates, although many groups are moving away from working with monkeys because they're just so close to us. But I want to make a distinction here. So recently, of course, we know that the Chinese doctor, who I'll just call JK because I have trouble pronouncing his name, did the first gene editing on human embryos. Not the first gene editing on human embryos, but the first to take those embryos and bring them and make human beings. That was done in 2018. So he demonstrated, he took it out of an academic exercise and made it pragmatic, and then we had to start figuring out what to do with it. This type of editing work is going to be extremely important moving forward. And now there is, it's been 7 years. Now there's people calling globally to start working, doing more of this work and learning how to do it with precision to correct major genetic errors in some of these embryos, which is huge. So Kathy Tai recently just announced her Manhattan project, really to start doing editing on these embryos that carry a serious genetic defect. That's huge. And of course, that's controversial. These are technology. Anytime you work with humans, we're held to the highest standards of ethics, transparency, etc. Even embryo screening, which you just mentioned, which is a diagnostic procedure, sequencing the cells, some of the early cells in the embryo, is controversial. But it's we did genetic screening on one of our children pre-implantation. We had all the embryos sequenced. I welcome personally that type of technology. I'm sure if my wife and I produced embryos that were defective, we would have welcomed technologies to do that genetic surgery and fix them so we could have our children. So these are very personal decisions around technologies that people have to make. These technologies are in early stage. We should be very careful using them and very transparent. But I think with more examples of doing this and becoming more comfortable and experienced and continuing to refine the tools, I think they will be used, more and more frequently. I want to make a distinction though with cloning, because I've become a huge advocate of cloning in the sense that I believe human clones will come in the near future. It'll just someone will cross that line, and that it won't be that big a deal once the dust settles, because we're not doing any genetic modification to, to that genome. We're not editing it. It's using embryological processes that were well developed for IVF. It's really just a little bit of lab work to clone an embryo. It's a little different, but we've cloned now, I think it's about 30 different mammals, cows, pigs, camels, etc. And so as we've gained more experience cloning other mammals, including monkeys, we're getting closer and closer to that line of cloning a human. And like the first IVF baby in the 1970s, I think once we cross that threshold and we start allowing a few more and then a few more and a few more, eventually cloning will be one of the reproductive options that people have moving forward. And what excites me about that is, as we kind of reuse a genome again and again and again to produce essentially identical twins, but that are displaced in time, you collect a body of experience, of lived lives, that I think become the template for evolving ourselves. Once we've done this a couple of times, we know what precision edits we might want to make to prevent cancer or live longer or have other features, because our biotechnology is going to race ahead faster than our generation times. So I find cloning to be super exciting.
Yeah. What do you think are the biggest bottlenecks, whether scientific or non-scientific? Like scientific being something we don't understand yet, non-scientific like human institutions, the fact that there's laws against these things, or we have ethical considerations. If we kind of break those bottlenecks, we make a ton more progress, or that will accelerate lots of other things.
So I get asked a lot about ethics, and I've always said the same thing, that ethics are a spectrum, and they're dynamic. They're different in different communities. They're different at different stages in life, and how a person's been raised. They're very dynamic and very personal, and so they're tricky to nail down. I generally work in the area of, can it be done? And if it can be done, it will be done. So you have to prepare for it. And then if it can be done, and it will be done, can it be done legally? And that's not an easy question, because again, the law is a spectrum as well. It's different in every country. It changes over time, etc. So right now we are extremely conservative when it comes to any human technology. I think it slows down progress in some way, particularly if we're restricting self-experimentation, because, I look at my genome and my body as my own to do as I wish, provided, I'm an adult and have informed consent, etc. Like I can understand the processes, and that others don't have necessarily a financial consideration in whether I make a decision left or right, and to go forward or not. So I think a lot of these institutions that regulate biology, that from and even fund biology, are very conservative right now. But I think that moving forward, they're going to change. And certainly, you're looking at, how do you create geographical regions with laws that are more permissive? I think that's huge. I think that we have to keep doing that. I think we have to build communities that are much more transparent among the community, because right now the big biotech players are all competing against each other, and they're essentially silos. And although they can do amazing things and they want to succeed, they don't necessarily want the same, for the other companies. And academia, which can open the door, can't develop the products all the way to market. So I think we have to change the institutions around funding, around data sharing, around publication, and of just, of results, positive and negative, what works and what fails as well. And ultimately just create new zones where we can be much more permissive for those that want to explore this type of engineering, including on themselves.
Well, for any long-term listeners of the show, they will know that this is why I'm doing what I'm doing, right? So to build these kind of regulatory zones to advance the science and to increase the speed of iteration, right? Because, as you said, existing institutions are extremely conservative, and that just means we get less data, right? So we can make progress less fast. At the same time, though, I'm also wondering um, how does what are the risks, right? So we can imagine, especially when it comes to this kind of technology, we've seen, for example, that a virus can escape the lab, right? So whether that specific virus that many of us are thinking about probably was actually in lab or not, let's leave that aside. But whether or not it's the case, that is a very realistic risk, right? And it's very hard to sort of design institutions for that kind of accountability. Like, again, whether or not it was a lab leak or not, like who would be responsible for it? Like everyone will want to deny it, and sort of questions around liability, whatever, they're almost like impossible to solve. But at the same time, these kind of risks could be existential, right? Sort of a more powerful virus, or so how do you think about sort of mitigating these kind of risks, and how does it square kind of with the idea that I also support, especially of very open kind of data sharing? The reason I'm asking is it's just something I haven't resolved in my head, but I kind of see and support the strong argument that the more we share data, the more we can collaborate, the more we can build safety mechanisms. At the same time, the counter argument is always, well, you don't want to give everyone the technology to build a nuclear bomb in their garage, right?
So this is tricky. And it's tricky for a number of reasons. I've started running into a wall with this. A couple of years ago, I championed an X Prize for next generation DNA synthesis and assembly. I wanted us to get to about 5 million base pairs of DNA, the DNA chain, so microbiology would essentially become programmable. And it was very well received and supported, except that the pushback was, this is going to accelerate the potential for bioterrorism because it would make it extremely easy and cheap to engineer a virus. And I couldn't disagree with them because it's true, it would accelerate that possibility. But my pushback to them was, we've had the technology to synthesize viruses, viral scale genomes now for over 20 years. And to make most human viruses, which tend to be quite small in terms of their genome size, that it's at a price point you could put on a credit card. And now with AI tools and just really simulators of, there's not that many formulas necessary to understand kind of the burst size and the spread of a virus in a population. The math is actually pretty simple. It's like you could design a pretty bad bioweapon and build it with today's technology. So why we need to start today, really, post COVID, certainly, where we saw the huge effects of a single virus particle on humanity. We need to build a new biosecurity architecture, and one that is not just company specific, one that is not nation specific. We kind of need to build a detection and remediation system, what you might think of as a global immune system, like our bodies have to detect and counter, threats. We need to build that for our planet. And that's going to take a level of cooperation that we've never seen top down with governments. And I think it's going to have to, really be created and be built and emerge bottom up by the folks that are supporting decentralized science and technologies. And that's where I'm putting my efforts today. Like I believe we have to build these decentralized biodefense systems, and that we're overdue. And it will probably take more another sensitizing event. COVID was bad, but not bad enough, and we did not have the transparent information we needed to determine the source or the dynamics, etc. So it just became the perfect breeding ground for conspiracies. I think we need to factor that in, and we need to make a completely transparent system. We need to start doing it now, because there will always be epidemic threats. And again, I'm not worried about bacterial epidemics. Those can be contained quite easily. I'm talking viral epidemic threats, either natural or potentially engineered. And we are so overdue. So I've been running a series of biosecurity meetings all year. I seeded at IndieBio last year. I would love to see a biodefense company come out of your organizations. I think it's crucial for the blossoming of this field, and ultimately to get to the endpoints of where we trust the engineering that is being done on any plant, animal, or person.
Yeah. And the argument that I always give to people in that context is, well, all the examples I can think of like existential risks from new technology that was developed, like whether it's nuclear or weapons technology, and even on the bioterrorism side, was actually developed by governments, right? And often like their militaries to get like a military advantage, right? So, the idea that we kind of restrict using these kind of institutions to restrict access is rather a recipe for an increased risk of abuse of these technology rather than the other way around. Sort of people always have this like James Bond villain scenario in mind of like some like private criminal gangs that band together and do these things. That is just not the reality. In reality, the kinds of villains who abuse these kind of technology are existing state institutions and governments, right? So,
True, but the thing to remember is today the foundational tools for doing this engineering are globally dispersed. They will always be accessible because they're essentially commodity. And a single individual potentially has the same capability as a larger group today, given the AI design tools and frankly the huge number of databases we can available today, including viral databases that we can train the systems on for doing the engineering. Plus, you don't need a manufacturing plant to build a pandemic. It is the host organisms and their intermixing that ultimately is going to cause the creation and spread of the particles. So you can seed it with a microtube's worth of viral particles. It is essentially one of the bottlenecks in our technological evolution, our biological evolution, that I think we have to get through, and where we just make it near impossible to harm each other with viruses, and of course we can counter anything that nature can produce.
Yeah. And so, final kind of set of questions. So you've also done commercial ventures and companies. So you're an entrepreneur as well. What advice would you give kind of an entrepreneur who wants to work in with synthetic biology, what the science has available, wants to commercialize it, as opposed to, or what advice would you give a scientist that wants to become an entrepreneur that wants to commercialize things, and how would that differ?
Yeah, so most of my experience in the world of biotech. I haven't made that many companies. The first company that I made was after I left biopharma, because I worked for one of the large global pharmas, and there I saw, what having huge resources in R&D can deliver. It's amazing, but I was watching our the outputs of drug development stay essentially flat. Like if you look at the amount of drugs approved by the FDA per year, it's always under 100. It's typically under 50. It just kind of floats around this mean, and that doesn't change despite huge amounts of investment in new technology. And so eventually this was described as the reverse of Moore's law, Eroom's law. And I think that, I wanted to get around that. So I incorporated a cooperative open drug development company called the Pink Army Cooperative. I built it in Alberta, Canada. It didn't work because I'm not really good at managing people. So I'm not good at running a democratic organization. Um, Ultimately, I ended up taking the same technology, the ability to write cancer fighting viruses, and made it into a standard C Corp. And that's operating out of New York City. It's called Humane Genomics, and they're doing well, but it's not open. It's not transparent. Hopefully they'll do okay, and we'll be able to beat the cancers that we target. I've also founded a global genome project, the Genome Project Right, which is still in its early days, but we are absolutely, as a species, going to start writing human genomes. And I hope we do it collaboratively and openly and transparently. But what I'm trying to do today is build open systems, because we have to share information. It should be as transparent as possible, in across the finances, across legal, across the scientific publications, across just knowledge in general. I think it's just going to be the huge accelerator. And what I keep in mind is that all biology, again, as I pointed out, runs on the same code. So why should we be proprietary when it comes to DNA? It just does not make any sense to me that we lock it down. We should be sharing most of this information as much as possible, for our businesses to be transparent. So I would really encourage people to build next generation businesses using architectures that are much more transparent, much more portable across countries, etc. Seek different sources of funding than just venture capital, where they really are much more predatory. Try and get patient capital, because some of the development timelines here can take longer than the standard 10-year VC fund for returns. And specifically, I try and get, when it comes to human therapeutics, focus on N of 1 therapeutics. And this is why if you start trying to make a mass market drug, you have the potential to harm millions of people. So you have to be extremely conservative in your drug development process. Also, it's a hard job, because you're trying to find the best fit for making a molecule or a therapy across a large population. So trying to find one size fits all is hard, but also getting through the regulatory barriers to maintain, to be able to make claims and also ensure safety is extremely hard. That's why we have the phase clinical trial. That's why we have Eroom's law. But if you go to N of 1, and we're seeing examples here, like with baby KJ Muldoon, that, you can make a personalized therapy today on the order of months, which is huge. And particularly for rare diseases in kids, this is a game changer. And for cancer, because cancer is ultimately a rare disease. No two people have the same cancers, because it's your cells that have corrupted that are now infecting your body. I think these are going to be the big drivers, and it's a complete game changer, because when you do N of 1, you can actually get approval to make the drug for a particular person before you even start making the drug. Because they've run out of the conventional processes. And of course, N of 1 is ultimately almost a form of community-driven self-experimentation. So I think there should be very, very lightweight regulatory around that, especially because you can keep it all transparent.
Did you say that already exists, kind of an approval process for N of 1 therapies, or does not?
There is an expedited process in the U.S. with the FDA. Every country has different rules and regulations, but I look at it this way: if I want to take a drug, I have some agency. Whether it's at a party, whether it's to treat my own cancer, and as long as there is transparency. As long as I am able to understand the risks versus benefits for myself and make that call, and importantly, no one, there is no economic incentive for another to ensure I take the drug. I think that is huge. Now, um, we don't have that as a generalized process yet, but I think we need that just for freedom to operate, for freedom to explore. And again, I think all the results that come back from this type of N of one development needs to be put into a meta analysis. So that we build a knowledge base of doing this type of work. You see this already with some of the ASO gene therapies that they're doing. That as the first one took a long time to do, and now we're getting extremely fast and turning them around with precision on the order of months. And with further experience, I think we can turn that around to days, maybe even hours. Because these processes are amenable to standardization and automation.
Mm-hmm. That's a very valuable insight. So if anyone listening is working kind of on personalized medicine or N-of-1 therapies, reach out to me. Because if you're listening to this podcast for the first time, in Prosper, we can basically develop regulations within a couple of weeks or months to make that possible and to make that legal. And also kind of the try laws that I've been involved with Montana, for example, that would be flexible to allow these N-of-1 therapies. And in the New Hampshire right to try law, they actually very explicitly allowed individualized genetic medicines or something like that. I don't remember how to word it. So on the regulatory side, there is opportunities opening up. So if you're listening and working with us, then reach out to me. That's a very big opportunity, I think.
I think N of one, right to try, even groups that are becoming more permissive around end of life will be at the forefront of developing these personalized therapies. Again, it's generally considered unethical to charge, to have a profit motive in clinical trial. But I think that just cost recovery is within reach, and again, the cost of developing many of these therapies is decreasing. And I just think this works in its scales. So I have no interest in making a one size fits all drug, none whatsoever. I want to see these processes be developed that can empower individualized medicine, because we all are born, exist, and die as individuals. It just makes the most sense to me.
Yeah, the paradigm is changing to predictive, preventative, and personalized, right? So
absolutely, yeah.
Mm-mm. Fantastic. Andrew, this was a really fascinating conversation. Is there anything else you'd like to give our listeners on the way, or where they can find you or learn more about your work?
I'm pretty transparent. I these days I do most of my work in communications. I will invest in a company if I see the need for it. I'm a early stage investor. I don't have a ton of money to do this. My next venture, which I've been exploring for a while, is looking at this future where cloning is going to just be available for people and become more accepted. I think this will happen over a generation, but to prepare for that, I'm exploring biological banking, coupled with financial banking and data banking, so that people can essentially back up themselves like we would back up a computer for eventual restore. I would love to see a lot more R&D go into things like cryonics and just being able to turn off for a while. We're going to need this for space travel or patient stabilization for transportation. I think that's a huge area of development, and I definitely want to have see a lot more R&D into understanding our brain and how consciousness is created in our neural material, and more than that, how memory is stored. This is these are all very early days in understanding the most important organ in our body, our brain. So hugely optimistic that the AI winter that we've been experiencing, the AI winter is over and we're booming. The biotech winter that we've been in post-COVID won't last too much longer. And when it starts to take off, biotech is going to have a sustained boom for decades. So really, really exciting times. Don't be afraid of biology. It's a lot more like engineering now.
Ending on a very optimistic note. Andrew, thanks so much for coming on the show.
Thank you so much, Nick. Really looking forward to getting down to visit you sooner than later.